what I think would be good would be for you to go ahead and explain,
Yeah.
Maybe in layman
to do
terms,
that.
but just kind of generally what it is that optogenetics is about.
Right.
And then you're talking about you don't know where RDoC is necessarily taking us. We're looking for the domains.
Right.
How might optogenetics
Well, that's a good
influence that?
It's a good avenue to think about. Okay, so remember what I was talking about is neurocircuits, right? So neurocircuits are groups of neurons that interact to achieve a task, often to guide behavior. And optogenetics is one technique. Optogenetics is one technique by which we can manipulate these circuits. So the technique of optogenetics relies on the identification of a number of different proteins from bacteria and other microorganisms that we can take out of those bacteria, the genes, and insert them into mammalian neurons. And those proteins are specialized to take light and turn that light into electrical activity. They can either be activated by light, such that shining a light on these proteins increases electrical activity in a neuron, or they can be in such a way that shining a light on these proteins decreases electrical activity in the neurons. And so these proteins, which we take from bacteria and we put artificially into neurons, give us the ability to manipulate with light the neurons that we put the protein into.
Okay.
So that's the basics of the optogenetic technique. Now imagine that we can just take those proteins and not put them into every neuron in the brain, but only a very specific subset. I mentioned earlier that we know the amygdala is important for anxiety and fear.
Mm-hmm.
And we can put these proteins into the amygdala, only into amygdala neurons. And then we can manipulate amygdala neurons. We can make them active, and what happens? We induce fear-like responses in mice. If we make them inactive, we reduce fear-like responses in mice. We can do a lot better than just that, because we can identify specific subtypes of neurons in the amygdala, and we can tag them with our proteins. Or we can look at cells in the amygdala that project to just one other brain region. Most cells in the amygdala, they project to lots of different brain regions. But what if we put our proteins only in those that project, say, from the amygdala to the cortex?
And that are only involved in anxiety?
Well, then what we learn is that different cells in the amygdala are involved in different behaviors. Even if you turn on the whole amygdala and you make the mouse anxious, if you turn on some cells in the amygdala, you make the mouse anxious. And if you turn on other cells in the amygdala, you actually make the mouse less anxious. And what we're learning by doing this on a very, very fine scale is that we can identify small subcircuits within places like the amygdala that are responsible for different parts of behavior, some of which make the animal more anxious, some of which make the animal learn something new, some of which reward the animal. And we can break apart behavior that way on a circuit-by-circuit basis. So optogenetics and other techniques like it, there are other techniques where we use other methods besides light to manipulate activity, but optogenetics and other techniques like it allow us to then not think of the brain as little parts, but think of the brain as circuits that are interwoven amongst those parts, with small groups of neurons in those circuits being incredibly important for specific behaviors.
It sounds like you might also start now coming up against some ethical dilemmas that we've never traversed before. You know, how might you deal with those?
So I think the biggest ethical dilemmas is I think before ethical dilemmas, there are technical dilemmas.
Okay.
How do we do those things in humans?
But if you get them done, then you'll have to face the ethical ones.
Right. Then I'm not sure what ethical dilemmas you're thinking about. I can think of a few. So one is, it's a little bit like mind control, right?
That's what I'm thinking.
If we're turning on and turning off specific circuits in the brain of a human, we could maybe turn on and off specific memories. What was the movie?
Yeah, it's Eternal Sunshine
of the Spotless Mind.
Yeah.
That comes to mind.
Yeah.
And in fact, in animals, That's one of the things that people have tried to do, is erase specific memories. Maybe it's worked, maybe it hasn't. But one could imagine doing that when you have this kind of exquisite control. I think that's, that could be problematic, and I would rely on my colleagues who've given these ethical dilemmas some serious thought to add their voices into the picture, and I think that's an important thing to do.
Yeah.
Most of the things that I've thought about doing with it, those ethical dilemmas have been raised and dealt with. And specifically, I'm thinking about things like, let's say, treating depression or anxiety. So I remember actually as a first-year medical student, and I wasn't interested in neuroscience at the time, I was interested in cancer. But one of my mentors that was assigned to me in medical school was a psychiatrist, and a group of us went to his house for dinner. We had a delicious dinner at his house, and he opened the field up to questions about psychiatry, because that was his area of expertise. And I remember I asked him a question about antidepressants, and isn't that really changing the way who people are when you're giving them a drug that affects the brain? And of course there are concerns. We don't want to make someone a different person by treating them with a drug for depression. And in fact, our antidepressants don't really change who people are. They release who they are by relieving them of the burden of their depression. But with these circuit treatments, do we worry that we might change who people are? Do we worry that we might muck with their essence of who they are? I think, yeah, it's a possibility, but in the end, it's really not much different from using any other treatment. We're talking about biology and psychology before. I think we should be more concerned about a psychotherapist's ability to change who someone is and whether that can be ethical or not. And there are some psychotherapies that people have tried that have been declared unethical, such as conversion therapy for individuals who are homosexual.
Well, and that is the kind of thing I was thinking is that, then if you cross those technical barriers and you become able to manipulate what a person can feel and how they respond to their feelings, wouldn't we then eventually have to have some codification of, well, this is what's good, this is what's bad, this is when it's implicated and when it's not implicated as an intervention? And yes, I realize that's a frontier we haven't gotten to, but it just seemed like it would be worth hearing your thoughts on it.
Yeah, that's interesting. It's, again, far behind sort of where I've thought, because I don't think we're there yet technically. When I treat patients, again, I'm going to go back to my own experience with patients on this. When I treat patients, I treat them because they come to me with a problem. They come to me because they're overwhelmed with depression, or because they're hearing voices that are scary or frightening or ruining their lives, or they're coming with intractable anxiety where they can't function in their workplace. To relieve those symptoms, there is no ethical dilemma in my mind.
Okay.
Right. So if we target our treatments towards that kind of dysfunction, I don't think we're in troubled waters. I can imagine that one could think up some diabolical schemes whereby we would use advanced technology, and most importantly here, without the patient's permission and guidance, that would be wrong. I can't imagine how you'd figure out how to do that, except through extreme force. But yes, there are ways that you could go beyond what would be treating a disorder. But frankly, I think those ways would be accomplished much easier with much less technology. Give everyone Quaaludes or something, I don't know. But I think the ethical dilemmas have been mostly dealt with in my mind, although, as I said, I look forward to hearing from my colleagues who've given this stuff more thought.